Good evening, kids. So, in yesterday's class, we have discussed at what is cell, how the cell is being discovered by Robert Hooke. When he has seen a cork cells under the microscope, we have seen the types of organisms based on the number of cells present in their body as unicellular and multicellular organisms. What is the living organism made up of? And finally, we have discussed about the structural organization of any cell found in the living body. While discussion on the structural organization of any cell, we have seen that any cell, when observed under a microscope, it is generally composed of three main components: cell membrane, which is also known as plasma membrane, nucleus, and cell wall, cytoplasm. So there are generally three components: plasma membrane, nucleus, and cytoplasm. But in addition to the cell membrane, plant cells also have a another covering outside it, which is known as cell wall. Then we have seen that what is the plasma membrane made up of and how it helps in the movement of substances in and out of it. Okay, while discussion on the movement of the substances, we have seen that there are two main processes through which the substances move in and out of the cell through plasma membrane, that is osmosis and diffusion. Okay, now in today's topic, I have sent you one video showing and explaining about the different types of solution which helps the molecules to pass through the plasma membrane. Okay, so we have seen that basically there are three types of solution: hypotonic solution, isotonic solution, and hypertonic solution. Okay, hypo means less, tonic means solute. So if the solution is having less solute concentration, that means less salt concentration and more water concentration, then that type of solution is known as hypotonic solution. Okay, as a result, what happens in this slide, I have shown you, I have taken RBC, that is red blood cells present in our human body. Okay, we have taken a red blood cell and we have put it in hypotonic solution. Okay, as I have already said you, and you have also seen in the video, hypotonic means less salt concentration and more water concentration. Okay, so as compared to the outside water concentration, the water which is present inside the cell is less. Okay, so as you all know that due to osmosis, what happens, the water from the outside environment will start entering into the red blood cell. You can see the yellow arrow, it is showing that the water will keep on moving inside the red blood cell because outside the concentration of water is more. As a result, you can see in the diagram that the RBCs are bursting. So the cells eventually burst because the osmotic pressure inside the cell increases due to continuous inflow of water. So when you take a RBC and put it in the hypotonic solution, it bursts. Okay. Next is isotonic solution. Isotonic solution means when the surrounding medium and the inside medium has equal concentration. Iso means equal, tonic means concentrations. Okay, so in the second PPT you can see that I have taken RBC and I have put it in isotonic solution. Okay, so the amount of water which is entering inside the RBC, that is yellow, it's being denoted by yellow arrow, and the amount of water which is being coming out of the RBC, which is being denoted by green color, both are equal in concentrations. So as a result, what happens, the water will keep on flowing in, moving inside the cell, and at the same time the same amount of water will keep on coming outside the cell. So there will be no change in size of the cell and the cell will remain as it is. Okay, and the third last type of solution is hypertonic solution. From the name itself you can make out that hyper means high, tonic means concentration of the solute. So when the concentration of the solute is very high outside, that means the concentration of water is less. So when you put, see here in the diagram, I have put RBCs in a hypertonic solution. So hyper means high concentration of salt. As a result, the concentration of water is less outside. So what happens, the water from inside of the RBCs will start moving out of the cell, will start coming out of the cell, isn't it? The green color arrow is showing the movement of the water molecules from inside the cell to the outside environment. Okay, as a result, you can see the cells have shrink, they have shriveled, and that phenomenon is known as plasmolysis. So when you And this process of movement of water outside the cell is known as exocytosis. Exo means out, cytosis means movement. So when you put any cell in a hypertonic solution, what happens? The cells shrink. They start shrinking because they lose water, and and this phenomenon of shrinking of cells is known as plasmolysis. Okay, so we have seen that water enters into the cell and water leaving the cell is respectively known as endocytosis and exocytosis. Endo means entering inside the cell and exo means leaving the cell. So endocytosis is nothing but it is the flexibility of the cell membrane that allows the cell to engulf or to ingest the food and other materials from the external environment. Okay, you can see the diagram. So when we have taken three solutions, hypertonic, isotonic and hypotonic. So when you are keeping the red blood cells in a hypertonic solution, it shrinks. That means the cells undergo plasmolysis. When you keep the cells in an isotonic solution, there will be equal movement of water in and outside the cell. So the cell shape remain as it is. It does not undergo any kind of swelling or plasmolysis. And in the third type, that is a hypotonic solution, since the concentration of water is more outside, it keeps on entering inside the red blood cells. As a result, the red blood cells burst eventually. Okay, so the processes by which the water or the food materials is entering the cell or leaving the cell is known as endocytosis and exocytosis. So endocytosis is again divided into two categories such as phagocytosis and pinocytosis. Okay, phagocytosis means when the cell is taking the food, solid food material inside its cytoplasm, then that process is known as phagocytosis. Phagocytosis is also used by the cells such as neutrophils, which is having phagocytic functions. That means if any microorganisms invade our body or any pathogen enters in our body, then the cells undergo which process? Phagocytosis. They engulf those pathogens and they destroy it. Not only that, you know that cell is a living cell, so it needs food to survive. So when it needs any kind of solid food substance, then also it undergoes phagocytosis. On the other hand, when the cell needs any kind of liquid material to be taken inside, then that process is known as pinocytosis. So endocytosis is the movement of the food, solid materials or any kind of food inside the cell, and it includes two categories, phagocytosis and pinocytosis. Okay, so here in this slide we will see through the help of a diagram that how it works. So in the first diagram you can see that yellow color covering is the nothing but the cell membrane, which includes the cytoplasm, and outside is the extracellular fluid. Those green color, red color are the food materials. Okay, or you can say any kind of material that needs to be taken inside the cell. So when it comes in contact with the cell membrane undergoes invagination, and it at a result it detaches from the cell membrane, and it stores that food material in the form of a vesicle. Okay, in that second figure you can see that the green color covering is nothing but the plasma membrane of the cell, and it has been shown that in animal cells endocytosis also takes place with the help of receptors. Okay, there are certain big molecules like proteins. So proteins are so big that they cannot pass the plasma membrane easily. In order to pass through the plasma membrane, they need to attach themselves with the receptor molecules. You can see those red color bilobed structures. These are receptors. Okay, those receptors identify the particular molecules, and then they get attached with those molecules and they push them inside the cytoplasm of the cell, and inside the cytoplasm they form vesicles, and those vesicles are known as coated vesicles. Okay, next you have seen that pinocytosis. I told you that any kind of fluid, when it is entering inside the cell, then it is said to be pinocytosis. Okay, and third type is food. If any kind of solid substances needs to be entered inside the cell, then that process is known as phagocytosis. Generally, phagocytosis is also taking place in case of amoeba. Amoeba takes its food inside its body with the help of phagocytosis. So when the food molecules enters inside the cell, it gets stored in the form of a vacuole, and that vacuole is known as food vacuole. Okay, so this is the different ways how the endocytosis is taking place inside the animal cells. This is another diagram which is showing phagocytosis, when the solid particle, it may be a food particle or it may be any pathogen, it gets engulfed inside the cell membrane, and this food vacuole is also known as phagosome because it is formed by the process of phagocytosis. Okay. Next is pinocytosis, any kind of fluid substance entering inside the cell membrane, inside the cell, then it forms the vesicle. And the third one is receptor mediated. With the help of receptors, the large molecules such as proteins and all entering inside the cell, and those form coated vesicle which is having coat protein outside its surface. So this is all about the types of endocytosis explained with the help of diagrams. Okay, and the last one is exocytosis. Okay, exocytosis, as I have told you, exo means outside. So cell is living, so it also produces harmful materials which needs to be excreted outside. Okay, and sometimes cells also secrete certain useful substances like interferons. Interferons are a group of proteins which is being secreted by the virus infected cells. Those interferons gives stimulus to the other cells, informing that the neighboring cell is being infected with the virus cell, so that they can start preparing its antibodies. So whenever the cell needs to secrete any kind of useful substance to the outside environment, or if it needs to excrete its waste material outside the body, outside the cell, into the extracellular fluid, then it undergoes the process of exocytosis. Okay, when the metabolic wastes are thrown out of the cell, then that process is known as aphagy. It is also known as aphagy. If it is harmful, if the waste material is being thrown out, then it is known as aphagy. Okay. So this is all about cell membrane and how the movement of molecules takes place through the cell membrane inside in the cell, and how the solutions helps the cell to change their shapes due to the movement of molecules, and how the different type of solid as well as liquid materials enters inside the cell by the process of endocytosis, and it leaves the cell by the process of exocytosis. Okay, again I have told you that in plant cells, in addition to the cell membrane, and another rigid non-living structure is present which covers the plasma membrane, which is known as cell wall. So cell wall is a peculiar characteristic of all the plant cells as well as fungi. Okay, they are not present in animal cell. Okay, it is generally made up of cellulose, and you can say that it is non-living. Okay, so it not only gives shape to the cell, but at the same time it protects the cell from the mechanical damage and infection. As I have already told you, since it is the extra additional covering of the cell, so it helps the cell, it protects the cell from the mechanical damage and infection. So below I have put some of the functions of the cell wall. So first one, it gives shape to the cell, you know very well. Next, it helps in giving rigidity to the cell, means a particular rigidity, shape, a perfect strength to the cell. Okay, because it is the additional covering. It gives mechanical strength. As you all seen that when a RBC is being put in the hypertonic solution, what happens? The water keeps on coming outside. Okay, but when a RBC is put in a hypotonic solution, then what happens? The water keeps on entering inside the red blood cell. So since red blood cell doesn't have cell wall, so it easily bursts. But in case of plant cells generally doesn't burst, because in addition to the cell membrane, they also have a cell wall which also helps in maintaining the osmotic pressure of the cell. Then it also supports the internal structure of the cell. It protects the cell from osmotic bursting, as I have already explained, and it also helps in the expansion of cells. Not only that, it also helps in cell to cell interaction. You must have seen that one cell interacts with the other plant cell through a connection which is known as plasmodesmata. So this cell wall also helps in cell to cell interaction. It also acts as a barrier for certain macromolecules, very big molecules. They cannot pass through the cell wall. That means it is how it protects the cell. In the last one in slide, you can see exocytosis, how it is happening. Any kind of excretory product or any kind of useful substance which needs to be secreted, it is being stored inside the cell in the form of vesicles. Okay, who prepare vesicles? Golgi apparatus. In the further classes we will learn the different type of cell organelles. In that cell organelles we will see that Golgi apparatus is one of the cell organelle which acts as a packaging material, means it helps in the packaging of different materials, like how we get food from the market packed in different packets, like Maggi in separate packet, Ippi in separate packets. Just like that, every material is stored inside the cytoplasm in separate vesicles. Now what happens when it needs to be excreted outside the cell? Then it gets attached with the plasma membrane. It fuses with the wall of the plasma membrane. As a result, those materials are being secreted into the extracellular fluid and then carried out through the blood and excreted either by kidney or by respiratory exchange. Okay, then we have seen, as I have told you, plasmolysis. Plasmolysis is nothing but the shrinkage of the cells. Okay, when shrinkage will take place? When you will put RBCs in hypertonic solution. Hypertonic means higher salt concentration and less water concentration. So as a result, the water from the cell will start coming out and the cell shrinks in their size, and this process is known as plasmolysis. In this figure you can see plasmolysis is being shown. We have taken bean seeds. In those bean seeds we have put sucrose. So sucrose is a salt, it's a kind of salt. So it becomes hypertonic. Since it is hypertonic, it means what? The water will start coming out of the cells. So the cell shrinks in shape and the plasmolysis is taking place. Okay. And you can see in plant cell what happens. The cell, the outer covering is cell wall and below it cell membrane is present. So when the shrinking is taking place, the plasma membrane shrinks and it starts coming away from the cell wall. This is how the plasmolysis takes place. And the last part is protoplasm. Protoplasm is nothing but it is the living component of a cell. You have seen that cell wall is non-living, but apart from cell wall, cell membrane is living, nucleus is living. Okay, so all the living components of a cell comes under the, or together forms protoplasm. It is translucent and it is a jelly-like viscous semi-fluid substance. Okay, and it encloses two basic structures. One is nucleus and cytoplasm. So we can say that nucleus and cytoplasm together constitutes the protoplasm of a cell. So this is all about today's discussion.